Welcome to 11th Ncert of Biology Unit 2 Chapter 6 Anatomy of Flowering Plants. You can very easily see the structural similarities and variations in the external morphology of larger living organisms, both plants and animals. Similarly, if we were to study the internal structures, one also finds several similarities as well as the differences. 6.1 The tissue. A tissue is a group of cells having common origin and usually performing a common function. A plant is made up of different kinds of tissue. Tissues are classified into two main groups, namely meristematic and permanent tissue, based on whether the cells being formed are capable of dividing or not. 6.1.1 Meristematic tissue. Growth in plants is largely restricted to specialized regions of active cell division called meristems. Plants have different kinds of meristems. The meristems which occurs at the tip of root and the shoots and produces primary tissues are called apical meristems. Root apical meristem occurs at the tip of root, while the shoot apical meristem occurs at the distinct-most region of the stem axis. During this formation of leaves and elongation of stems, some cells left behind from the shoot apical meristem constitute the axillary bud. Such buds are present in the axilla of leaves and are capable of forming a branch of flowers. A meristem which occurs between mature tissue is known as intracalary meristem. They occur in grasses, regenerate parts removed by the grazing herbivorous. Both apical meristems and intracalary meristems are primary meristems because they appear early in life of plant and contribute to the formation of primary plant body. Meristems that occur mature regions of root and shoot of many plants, particularly those that produce woody axis and appear larger than the primary meristems, are called secondary or lateral meristem. Following the division of cell in both primary as well as secondary meristems, the newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed permanent or mature cells and constitute the permanent tissue during the formation of primary plant body. Specific regions of the apical meristem produce dermal tissues, ground tissues, and vesicular tissues. 6.1.2 Permanent tissues. The cells of the permanent tissue does not generally divide further. Permanent tissues having all cells similar in structure and function are called simple tissue. Permanent tissue having different typical cells are called complex tissues. 6.1.2.1 Simple tissues. A simple tissue is made up of only one type of cell. Various tissues in plants are parenchyma, collenchyma, and sclerenchyma. Parenchyma forms the major component within the organism. Collenchyma occurs in layers below the epidermis in the dicotyledon plants. Sclerenchyma consists of long, narrow cells with thick and ligand cells having a few or numerous bits. 6.1.2.2 Complex tissues. Complex tissues are made up of more than one type of cells and these work together as a unit. Xylem and phloem constitute the complex tissue in plants. Xylem functions as a conducting tissue of water and minerals from the root to the stem and leaves. Tracheids are elongated or tube-like cells with thick and ligand-walled tapering ends. These are dead and are without protoplasm. Vessels is a long cylindrical tube-like structure made up of many cells called vessel membranes, each with ligandified walls and largely central cavity. Xylem fibers have highly thickened walls and obliterated central lumens. These may either be separated or aseptate. Xylem parenchyma cells are having a thin wall and their cell wall are made up of cellulose. 6.1.2 The tissue system. We were discussing types of tissue based on the types of cell present. Let us now consider how tissue vary depending upon their location in the plant body. 6.2.1 Epidermal tissue system. The epidermis is outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. Epidermis is usually single-layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and large vacuole. The cells of epidermis bear number of hairs. The root hairs are unicellular elongations of epidermal cells and help absorb water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. The trichomes is a shoot system, are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes helps in preventing water loss due to transpiration. 6.2.2 The ground tissue system. All tissue except epidermis are having and vascular bundles constitute the ground tissue. It consists of simple tissue such as parenchyma, collenchyma and sclerenchyma. 6.2.3 The vascular tissue system. The vascular tissue system consists of complex tissue, the phloem and the xylem. The xylem and phloem together constitute vascular bundles. In dicotyledonous stems, cambium is present between the phloem and xylem. Such vascular bundles, because of the presence of cambium, possess an ability to form secondary xylem-phloem tissue and hence are called open vascular bundles. 6.3 Anatomy of dicotyledonous and monocotyledonous plant. When we see a transection of a dicot plant and transverse section of monocot root, dicot root and monocot root, we see root hairs, epidermis, cortex, endodermis, pericycle, protoxylem, metaxylem, pith and phloem. When we come to monocot, we see root hair, epidermis, cortex, endodermis, pericycle, phloem, protoxylem, pith and metaxylem in sequence. When we come to see a transverse section of stem of a dicot plant and a monocot plant, first of all we'll see a dicot plant. When we see a dicot plant, we see epidermis, collenchyma, parenchyma, endodermis, pericycle, phloem, cambium, metaxylem, protoxylem and pith. When we come to a monocot plant, we see epidermis, hypodermis, vascular bundles, phloem, xylem and ground tissue basically. When we come to next part, that is 6.3.4 6.3.5 dorsiventral or dicotyledonous leaf. Next is 6.4.1.2 Activity of cambial ring. The cambial ring becomes active and begins to cut off new cells both towards the inner and outer side. The cell cuts off towards pith, matures into secondary xylem, and the cells cut off towards periphery mature into secondary phloem. The cambium is generally more active on the inner side than the outer side. As a result, the amount of secondary xylem produces is more than secondary phloem and soon forms a compact mass. The primary and secondary phloems get gradually crushed due to continued formation and accumulation of secondary xylem. The primary xylem, however, remains more or less intact in our xylem. The primary xylem, however, remains more or less intact in our around the center. At some places, cambium forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in the radical direction. These are the secondary medullary rays. You can see figure 6.9 in 11th Ncert. Then we have to simply summarize the definitions and simple definitions of springwood and autumnwood, heartwood and sapwood or cambium. Next is 6.4.3 secondary growth in the roots, and we have finally the summary, that is in figure 6.1.1 we see some different stages of secondary growth in typical dicot root, and there we finish. Thank you guys. Let's meet in the another video. Thank you.